We saw all of this yesterday. However, we can see a little bit more of what I was talking about of poor data quality in this set than we got out of the other sequencing. So again, 100 base pair sequencing, but the tail goes way down this time, right? So there's going to be a lot of reads that you're going to trim at least 2 or 3 bases off of, but probably a great deal more because the quality is poor. Everything else looks more or less normal for when this was sequenced. If you do a lot of NGS sequencing work, you will start to recognize which version of the Illumina chemistry got used by things like little dips, among other things. So it's an unfortunate state of affairs. Again, what you see here is a little bit different, right? This is a poorer quality sequencing run, and you can tell because there is at least a small uptick of reads, whole reads that are averaging quality 2. So if you have a whole read that averages quality 2, how useful is that read? It's the equivalent of random number generation, right, for sequence. So it's going to get discarded because the whole read is nothing but a 1 in 4 chance of being correct. So we're going to need to trim all that stuff. We have, again, the same kinds of noise in sequence that we had before, where there's a lot of junk at the beginning and everything else couples pretty closely to where it's supposed to be. I have never once in my life, by the way, seen a FastQC report where this isn't at least questionable. The expected normal distribution of G to C is almost never matching or within the expectations of a normal curve. It's just an unfortunate side effect. We don't have a lot of sequence bias. There's no ends that are showing up at particular positions. There's a slight uptick at base 5, but other than that, everything looks pretty normal. Length is always 100 bases for these. There's a little bit of sequence duplication, and look, it's almost all, or it is all, the true-seq adapter. So that's good. That means that when we get rid of it, everything else should be happy. There's adapter at the end of the reads, et cetera, et cetera, et cetera. So it looks pretty normal. We can take a look at read 2 as well. And it's going to look pretty much the same, right? The tail comes down. It actually comes down a little bit less fast, but it still comes down to poor sequence quality. All of the data look about the same for GC content, length distribution. No overrepresented sequences, but we are finding adapter content at the end. So again, all of this points to we obviously have not trimmed this data yet. So let's go ahead and trim the data. So this time, instead of using FAQCS, we're going to just use Trim-O-Matic to do the trimming. Uh-oh, there's a typo. Hold on. There's an extra line in there, I think. Let me try it before I say that. Yeah. It looks like there's an extra line. I can't tell whether it's actually a carriage return or something weird in the formatting. So let me... Well, if that's really an extra line, it's going to be a problem. If it's just formatting, then... Oh, come on. I don't want you there. Go away. There we go.
It's working fine.
Okay. Oh, well, it's not quite working fine. There is one more typo. So let me fix that, and then everybody reload the page, and you can copy and paste again. There isn't really any other way to fix it other than to paste it into a text editor. So let me... The disadvantage of me copying and pasting at 9:30 at night is that sometimes things get messed up. Okay, so reload your page and recopy that. The reason I say that is that what's missing off of that is the final statement, which is I'm going to go ahead and type it in manually. You can as well if you really want to, but it's cutting off at the minimum length of 25 for this sample. So again, it runs really fast for just a few for just a few reads. This is going to be directly proportional to how many reads you have in your sample. But we started off with 100,000 read pairs, got rid of 5,000, so we dropped about 5%. The vast majority of what was dropped was the reverse read. You see that here where the forward read is surviving. The reverse read is almost always the one that's dropped. And in reality, both pairs only dropped 69 times. So that means that in cases where paired reads where one had to be dropped, the other almost always survived with a very small subset of data. Again, there's so much more data in your paired read sets. There's very rarely going to be an occasion to use your surviving unpaired reads. There are people who would tell you to do so. I won't argue with them, but it's only 5% of your data. So have a good reason to use it because it complicates your analysis rather significantly in terms of what commands you have to run if you're going to add new data in. Okay, so we trimmed the data. Hooray. Let's go ahead and do it all again, except this time let's do it in a shell script, sort of like we tried to yesterday. So we're going to make a new file, and then we're going to paste all of this stuff in. So in this case, this script is only generating the data and trimming the data. I'm separating things out just so you can see what the separate programs are. But what we have is 6 gunzip commands. Again, this is the stuff that you're never going to do with your data because this is the subsampling so that we can get through the top hat later and finish on time. And then it's going to trim each one of those replicates individually using just trim-o-matic. And then when we've got it all pasted in, you can Control X, write it out, and we have a file that we will now bash. While we're waiting, a couple of other things to bear in mind. I'm going to take a little bit more time today to talk about using the command line and using Linux, since the actual stuff we're doing is a little bit less extra data. So actually it's a good spot because that's what I was going to talk about. Bash, which is the command that we're using, is I think the best way to think about bash as you're first starting to learn it is that it is like a tiny child. When it is doing what it's supposed to do and it's happy, it doesn't make a sound. It does not say anything. You don't get any complaints. It just does what it's supposed to do. When something goes wrong, it complains loudly and vociferously, and you're never going to get it to be quiet until you fix the problem. In this case, when I was trying to run it before, it was complaining. It had a whole bunch of weird stuff that didn't look like any of the output I expected, and it was because it couldn't find files because there was a space missing between file names, and they became one big long file name that it couldn't find. So, for those of you editing at home rather than me fixing I will go ahead and go fix the copy and paste. But in the meantime, all you need to do is go into Nano and add a space at the end of the first Java line between the GZ and the backslash, and save it again, and it will work when you try to bash it. So I'm going to pause there, let everybody run bash until it works, fix my error. So all of this, right, was, again, our attempt to make a bunch of work go faster by putting it all into a single command file. The command file that I gave you in this case is not complete. It doesn't have the module loads. It doesn't have bin bash at the beginning of it because we're just trying to get through getting the data. So now we have all of the data that we want. With no fast QC. So mail rep 1, read 1 and read 2, and then the QC version here. So again, with the naming conventions, right, what did we do? We took mail, replicate 1, read 1, fastq.gz, so this was our original input. We did QC on it, so our output became .qc.fastq.gz. So now we have a series of each one of those types that have been trimmed and not trimmed in our directory. Hooray. So what is this doing? And I had this a little bit earlier in the text. In terms of what's happening here. So I mentioned yesterday that Trim-O-Matic can do the trimming, and given that it's always easier to only use one tool instead of two, for future work I would say go ahead and use just Trim-O-Matic, and it's getting easier to use every day, so that's fine. We use the same Illumina clip tool that we used before, which is getting the adapters off. Note here that we knew that these were true Seq3 adapters. That's the one that we used. So this is how we knew which one to get rid of. There are other adapter names. When your sequencing is done, you can ask them what they used and make sure that you're turning off the right one, or you can identify it from FastQC. After it does this, and the other important thing to note is that Trim-O-Matic does these things in the order that they're written. If you put min length first, it will check the length of the read before it starts trimming and never again. So it does this in order. So first it does the Illumina clipping, and then it does this leading trailing. So the leading is going to be on the 5' end, trailing is going to be on the 3', right? It is checking for quality at or below 2 and removing any bases at the beginning, any bases at the end with a quality at or below 2. After it's done with that, it does a sliding window trim. I mentioned this to a couple of people. I can't tell you which trimming I like better. Well, I can tell you which one I like better because it's more straightforward and more understandable. There are multiple schools of thought on how read data and high throughput read data should be trimmed. The easiest and most straightforward is to just do a hard clip, which is what they call this, or a straight clip, which is saying it's going to start at the beginning. If the beginning has a quality of 2, it'll trim that. It'll go to the next base. If that has a quality of 2, it'll trim that, on and on, until it gets above 2. And then it stops. Do it the other direction. Sliding windows is taking exactly that. It's taking the average of, say, 4 bases. And if the average of that is 2, then it clips off all 4 of those. And then it goes to the next step. And if the average of that is below, it clips off all of those. It slides the window along until there's no more bases to clip. Either one of them is acceptable and used by many, many people in either direction. I can't tell you why anyone has their own preference. I prefer to just do leading and trailing. My personal opinion. I can't back it up with any facts or really the vigorous studies as to why it would be better or worse to do that than any other type of technique on any given sample. By the same token, I can't tell you that there is a good study out there showing you the value of or the quality of result for RNA-seq genome assembly variant detection based on what quality you trim at. There's a little bit of hand-waving and mysticism about what number you should pick to trim at because nobody has the time to do a really complete study to tell you what the best options are. And even if they did, the next person would come along and say, well, it's a new chemistry now, so we'd have to do the study all over again. Things are moving too quickly, so we're just going to pick a number that we like and get rid of it. Generally, I use sort of basic jump metrics. I start at 2. If I just want to get rid of absolute junk, 2 gets rid of the junk. 5 is the next step, 10 is the next step, 20 is the step after that. Don't go over 20. Right? So somewhere between 2 and 20 in increments of whatever you like is probably a good number to use. Your mileage may vary. You may trim differently and get different results. I'm sorry. Welcome to bioinformatics. Okay, so we've trimmed now, and the last thing it does is after it does all of that trimming, it checks, and if the minimum length of the read, if the length of the read is now less than 25, it throws the whole read away. So we've done that with all 4 of our samples. Now we are going to do the next thing, which is pretty much the exact same thing that we did yesterday, except. So It's these commands. You can go ahead and copy and paste them in. They should work fine. I believe I tested them yesterday. I hope I tested these versions of them yesterday. And it's going to be running Top Hat pretty much the same way as it did yesterday, except if you remember from yesterday, when we run Top Hat with one set of reads. Right? One sample at a time goes through Top Hat. How many sets of reads are going through Top Hat this time? More than one, right? It's all of them. All of the QC'd reads that we just generated are going through Top Hat. So why are we doing this? What are we doing that's so different that we're running all of the reads through Top Hat? So again, we're working with a semi-model organism this time. The gene model is really the major difference at this point between a semi-model organism and a model organism. Genome assembly is not a solved problem by any stretch of the imagination. However, it's really, really good. People are really, really good, and the tools are really, really good at putting a genome together with the associated reads that they need. What they're not so good at is figuring out what a gene is in a new organism that hasn't been studied very heavily. Before the human genome was finished, there was a multitude of studies about individual genes that they were really interested in. They knew more or less what an intron was and what an exon was and what the actual signals that caused those splices to occur were. So they had a really easy time, which was not particularly easy, but a much easier time of building a gene model for the human genome or the mouse genome than they would for, say, a chicken, where there's a lot less work that's been done on the organism to predict what genes should be. So we have a good reference genome. We have a fair to poor annotation. So what are we going to do here? We're going to map all of the reads to the genome using the fair to poor annotation. So why are we mapping all the reads here? So we can't annotate with reads. Reads don't help us annotate anything, right? All they help us do is, if we're taking RNA-seq, they help us find where genes actually are and aren't. So what's going to be missing out of a previously done annotation is better, or previously done gene calls, is more data. So the data is reads from RNA-seq. What are we looking for? We're looking for more genes and better genes than what we have in this original call. So what are we going to want to use to get as many genes as possible? As much data as possible. We don't care what sample it's from. We don't care. As long as it's from a chicken, you would take as many reads as you can possibly get that haven't already been used to annotate the chicken genome to build a better annotation. Right? What extra value do we have here? Well, this is from a blastocyst study, so it's a developmental study. How many other developmental studies are there likely to have been before this? Probably not many. So all of the genes that we're interested in a developmental study may never have been annotated because they would never be expressed in a full-grown chicken. So anytime you're working in a new tissue, you might want to do this anyway because there might be new genes that you've never found, right? Especially in a semi-model organism, but even in a model organism, you may find whole new genes. But that's what we're doing here. So we're taking all of the reads that we just generated, and we are going to align them to the reference, and we're going to use that information to build a better gene model. Right? All we're doing is aligning our reads to the reference using the current gene model. So that's going to allow it, where we know what the genes are, it's going to do a really good job of aligning those reads to the genes. Where we don't know where the genes are, it's going to do a slightly less good job, and it's working on it right now, trying to get all of that information output. Okay, so again, because it's Top Hat, we just made an output directory called Top Hat All. We're going to look at the alignment summary. Try and move it to the top of the screen so the people in the back can see. So, what do we have? Well, we had 380,000 reads come in, of which 270,000 aligned. 75%. So yesterday with model organism, we had about 90%, right? With a semi-model organism, we have 72%. Are we happy with that? Maybe. Probably. Again, we'd never be sure, but it's pretty likely that the chicken genome still needs a lot of work. So there are probably regions that are missing, and those regions might include coding regions, so we might have reads that are just not showing up yet. You could spend a lot of time trying to improve the chicken genome, but I don't think it would help you if this was your study. So you can take this information and go with it. The last bit of information. Of these, 2.5% had multiple alignments. What does that mean? Splice variants is one option, right? Because you have multiple copies of the same gene, so that's one option. What else can you have that would cause one read to show up in multiple places in the genome? Gene duplication, repeated sequence within genes, highly repetitive sequence, right? There are motifs that have to show up in every transmembrane domain, and so there are going to be reads that show up in multiple places. This is only really a problem if it gets really, really high, right? If all you have are genes that are showing up in repeated regions in the genome, you're not going to be able to get any useful information out of that because all those genes are going to be the same. So we're more or less happy. So the other bit of this to bear in mind is that the reads align 72% of the time. Because the gene model is not great, and possibly because the reference isn't great, some of those reads didn't align in what they call a concordant fashion. You have reads that you know are expected to be pairs. The tool expects those reads to be paired. When you say it's paired, what does that mean? Well, it came from a single fragment. The fragment should be maybe 500 base pairs long. So at best, you'd expect it to be about 500 base pairs apart, right? Not only that, but you'd expect them both to be pointing towards the middle of the reference. If that isn't the case, if one aligns here and one aligns way over here, that's a discordant alignment. If they align and they're both in the same orientation, that's a discordant alignment. All of these things are things that are checked. So 6% of the reads aligned, but didn't align in a fashion that made sense to the aligner based on the fact that they were paired reads. Some of that can be sequence error. A lot of that I would expect to be missing gene annotations because I didn't know what to do with those alignments. So we're going to do something now that's going to hopefully improve the alignment. So let's look at what we've got to do. So now we have a new accepted hits file that came out of this. So we're going to use a different tool. Yesterday, we stopped using the top hat cufflinks cummerbund pathway at top hat. Now we're going to use some of the other tools. We're going to use cufflinks and cuff merge to build the new annotation. So cufflinks is going to take the BAM file and turn it into a new GTF file. So cufflinks is making new annotations based on an alignment file. That's what all of this is going to do. So let's see if it works, shall we? It's doing something. And look how nice it is. It tells us how long it's going to take to finish, or at least how far along it thinks it is in the data set. That getting data is definitely something that everyone in this room should be interested in, references and all of that. I'll take a minute, since it went back down to 0%, and talk about two of those things. One is you're all taking this class through ICER, which means that hopefully you're all at least considering using ICER HPC resources to actually do your analysis. There is a directory on ICER HPC called Mount Research Common-Data. There's a subdirectory there called Bio. We tried really hard to deposit things that we know people are going to use a lot into that directory. So a lot of things like the MM10 reference and the human genome reference should already be there. You shouldn't need to download those again. You should be able to just point to that location. If it's not there and you think that it's something that would be commonly used, you are welcome, and please do contact us directly. Ask us to put it in place. It doesn't actually take us that much time. You're just not allowed to put things there because we don't want you to accidentally mess up our whole directory structure. So please feel free to ask us to download data sets and put them in place. Given that we are all busy, if you can give us a link, which we'll talk about how to find things in just a minute, that would be much more helpful so that we can actually make sure we're grabbing the thing you're asking for without having to do a lot of digging. Even myself or Duranya being biological experts in the domain of bioinformatics doesn't mean we know everything about your organism or your projects. Please assume that we need at least a little bit of a pointer as to what that's going to be. Given that this is taking longer to run than I expected, let me talk about references then. So we'll start with where to get your reference genomes, right? Because that's the majority of it. Your data itself, you're going to be downloading from a sequencing center mostly. You're not going to be mining for that. So who knows where reference genomes are deposited? Who wants to try to use NCBI to download reference genomes? Genome browser is another one. I say that sort of tongue-in-cheek. NCBI is, in my mind, in a lot of cases, the equivalent of a deposit-only repository. You can get data back out of it, but it's really difficult to do so, and the structure is not really conducive to making it easy. On the other hand, the Europeans seem to think that data should be easy to find and download. So, the European Bioinformatics Institute has a really fantastic structured website that enables you not only to find what you're looking for, but then to take those links of what you're looking for and type it into a command to just get the data that you want in the structure that you need it to be. So if we wanted to, say, look for... So I typed in Gallus Gallus because I want a genome. And what do I get? Well, I get a whole bunch of ensemble genes individually, but I want the whole genome. No, where did I go? Because I'm an EBI. EBI is for... Oops. So Ensemble is the actual genome browser portion of it. They're related to EBI. So, oops. Maybe if I put it over here. So here you can grab most genomes that they have deposited. So, chicken. You can go to the download, and I want to download so you can actually go through and look and say, oh, look, here's all of the information I want for any given organism. They have the GTFs, the BAMs, the bigwigs, all of the regulations, if there are any. You can search through this, or I can actually click on the organism and find in here the link to download all of the bits and pieces that I need. I really highly recommend using Ensemble. It's much easier to navigate. It's not incredibly difficult to understand what you're doing, and everything is intuitive and takes you to a place that makes sense. I frequently end up clicking through links in NCBI that take me in a giant circle of 3 steps, and none of it is useful. There are back-end ways to get into that data. There's the FTP repository where you can actually download it directly. But Ensemble is a much easier place to download the genes, the cDNAs, the proteins, the full genome, everything else that you could possibly want are all linked. You can grab them individually. You can go to the FTP site and find the locations. Variants, etc., which if any of you take Duranya's variant analysis class, you'll understand why DB SNPs for model organisms are useful, or at least hopefully she'll talk about it some.
So the data download for Ensemble is directly used by the whole time? top hat to do alignment?
Yeah, so
Then if you organize some format, right?
yeah, I'm going to take that and turn it around and say this. There are standard formats for all types of biological data. Genomes need to be deposited as FASTA files. Annotations need to be deposited as GTS, GFF3s, or GenBank files. Usually they're deposited as all three, or if they're not, it's a simple matter, and the site itself does its job of making sure that all three of those formats are available. When you download them, if you download the whole directory from Ensemble or the whole, there's usually a tarball somewhere, I have to find it, the whole zip file of it, it will put it into a directory structure that most tools use. In the case of Top Hat, right, we have to actually specify the full path to the location of the file. So it doesn't matter where you put it, just that you have it and that you have both of those available so that it can actually find them. But yes, the GFF file is a standard format. The FASTA file will be in a standard format. If you need any other annotations or other bits of information like the cDNA library or anything like that, those will also be in FASTA formats for whatever you need them for. Nuts and bolts. Okay, so let me get to nuts and bolts so I can talk about it. Which one are you talking about here? The reference of the top transcriptome index? Oh, that. Okay, so this is something, if it's not there, what this is this is a, I'll show you. It's probably easier to show you than to try to explain. So let me try to do that. So in that directory, the link that we were looking at here is reference, the Gallus reference all the way to the Bowtie2 index genome. So when you give it, if you give Top Hat the location of a FASTA file for the index, or what it's asking for is where is your reference, right? The first thing you have to give it after all of the options is where is the reference file, and then where are all the reads files that you're going to line it to. If you give it a reference to just a FASTA file, it will build this index. If you give it a reference to a pre-built index, which in this case is genome, and then everything after that is something that was built, it will just use the pre-built index. Okay. So you don't have to have that formatted the way that it is there. If you give it a link to a FASTA file with your reference genome in it, it will build a new reference and use it. Okay, so that's the quick, and we'll talk more. I do want to actually spend some real time talking about how to find reference genomes, what you need out of the reference genomes, and all that on Ensemble. But in the meantime, my run finished. So I want to look at it for just a second, and then we will. Yeah, it'll say that. Again, it complains, but in those cases there are warnings. It's saying, so it's trying to build a couple of things at once. Cuff links is trying to do a lot of work. Some of the work we don't actually need it to do right now. You might use it for other things. And one of those things is it's trying to, based on the reads that aligned, it says, well, I know that I aligned to, in genome, the chromosome reference at this point. And it's trying to actually rebuild a new FASTA of all of the individual genes instead of a reference genome, but the actual FASTA of individual genes. And to do that, it's trying to use the reference genes, but it can't find them. We don't need to worry about that for what we're doing. If you were trying to get those genes out, you'd probably want to go back and figure out what went wrong, that it couldn't find them so that it could correct it. But in the meantime, this is what a GTF file looks like. Let me try that again. every one of these lines we could open up in Excel a little bit easier is what the chromosome position is, how the call was made, what kind of gene it is, what the coordinates of the gene are, a whole bunch of information, whether it's a positive or negative stranded gene, whether it's on the plus or the minus side of the chromosome, and then all of the information that it can transfer over about what its name is and what its transcript ID is, etc. This is really great. There's a problem, however, which is one that is really dumb because it's part of the same tool set. The output from cuffdiff as it is, or cufflinks as it is created, is not compatible with TopHat. I'll let that sink in for a minute. Same people developing both packages developed a tool to build an annotation, and then another tool to do alignment using an annotation file, and then another tool to create annotation files that's incompatible with the tool that they built themselves. Welcome to bioinformatics. So what that means is that we need to get rid of some junk. Titus wrote a really nice script. We're going to download it. If we copy and paste these two in, it's going to grab that file, and then we're going to actually apply it to get rid of all of the no-strand information, all of the no-stranded gene annotations out of this file. So what we did was we took this merged output, which is what came out of all of that computational work that we just did. We applied this Python script that gets rid of all of the no-strand annotations and created a new file called nostrand.gtf. So cufflinks is taking your acceptedhits.bam file, which is the alignment of your reads to the reference genome, and is generating a new annotation. So it is taking just based on the alignment, with no information about the actual current gene calls, it is making brand new gene calls based on what reads you aligned to the reference and where that indicates that there might be genes present in the sample. And it's taking information about the start and end of alignments and using that information to call new genes. So that's what cufflinks is doing. Cuffmerge is then taking the output that just generated and merging it with the original gene calls and trying to overlap those, make sure that it corrects things and everything else to make a brand new gene annotation that is a combination of the original gene annotations and the ones that were just built using the cufflinks output, or the TopHat output. And I was trying to show, it's really hard to see in this file, but if we here, right, the gene ID is what it's essentially being made up by cuffmerge, but it's also making sure so that where there's an old here, it said, oh well, we found an old gene ID of this from the original from the original gene model. So it's going to be using the gene ID all the way through for this analysis, but it's also going to inform you later when you want to annotate your output that you don't need to re-annotate this gene because you can pull the gene annotation from your old gene file of NM205429 and see what it was. Old cuff 1.2, so what's the likelihood of that, right? You're merging two GTF files. So the cuff merge is saying, well, where did this annotation come from, essentially? So if both of them exist, I don't know how it decides which one takes precedence. But it may actually display both, or it may only display the one from the more authoritative annotation. So I can't tell you because again there's a lot of data here. My expectation, let me see. I haven't actually thought about that. Is the short version of why I'm being a little bit hand wavy. But let me look at the command here real quick. Yeah, so it's just trying to merge those two together. Where they both exist, it should either tell you that it existed in both originals, or it will tell you only one of those two is the old. I'm not sure when it would tell you which is which. Have to look into that a little bit farther. Okay, so we now have this new GTF file. We fixed it so that it is no longer broken. So we've been at this for an hour. I think we can go ahead and run get this started. Again, the transcriptome index is going to be the same, but now we're using this new GTF file, same reference file, but only one sample. So now we're going to start doing the actual analysis. All of that work that we just did, that first hour of time that we just spent, was just spent building a better gene model so we could do actual work. So let's go ahead and get that started. It should take 5 or 6 minutes, so if you want to take a 5-minute break, I'll walk around and try to help. If you want to go get stuff, I won't start talking for at least 5 minutes.